Hi, my name is Leo DePere, and I'm an AI researcher at the Califano Lab at Columbia. And today I'm introducing GREmLN, which actually just came out yesterday. And GREmLN is the first transformer-based foundation model for single-cell genomics that also incorporates graph information, or gene regulatory networks, in its inference to create better gene-level and cell-level embeddings for downstream tasks. And those downstream tasks can be anything from perturbation prediction, cell trajectory prediction, or further therapeutically relevant tasks. So one of the main issues with single-cell RNA-seq data is that there is no inherent structure or ordering to the genes. And that's something that inhibits transformer models because they really benefit from meaningful structure in the tokenized data. And so what we've done with GREmLN is we're actually using the gene regulatory network to inform a certain relative positional encoding for each of the gene tokens. And we do that by essentially altering in the transformer block, we just alter the attention mechanism such that the query vector is now being transformed to reflect that graph information. So we do that via a graph kernel diffusion method that we developed. And essentially what that does is it gives a soft inductive bias to the model. So we're feeding it this prior, the graph, the gene regulatory graph. And the model can use that because it's in its query vector. However, we're not altering the key. And so that means that we can learn at the same time local interactions between genes from the graph, but also long-term dependencies that aren't necessarily captured by the graph. And that's through the key. So essentially what we've done is we've trained it on mass language modeling, hiding the expression of 15% of the genes and asking the model to reconstitute those. And after validating that it understands the transcriptional landscape, we moved on to benchmarking. And in benchmarking, we both tested the cell level embeddings as well as the gene level embeddings. And on both of these cases, we also looked at unseen data with seen cell types and unseen data with completely new cell types, also from cancer, which we did not train on. And in each of these cases, we outperformed the other foundation models in the space. And we do that with only 6 million cells in our training set and 10 million parameters in our model, compared to roughly 30 million cells in the next smallest data set that was used by other models, and 30 million parameters by those same models. So for future directions, we're going to look more into perturbation prediction and the cell trajectory prediction, as well as some others. But in the meantime, we encourage you to check out GREmLN or just our preprint that's out, and also you can use it and play with it on the virtual cell platform, which you probably can't see from over there, but we do have a QR code that works. And real quick, I forgot to mention, but this was a big effort from both the Califano Lab at Columbia University and the Chan Zuckerberg Initiative, and specifically the Biohub in New York. And yeah, so I'd like to thank everyone on the paper for their collaboration and help.